Good morning or afternoon, everyone. This is Carl at NAI Central. And welcome to the latest in the Director Seminar Series. Our speaker today is Steve Daunt from the University of Rhode Island, PI of our alumni team, and he is speaking to us from the Marine Biological Laboratory, one of our current teams. So we have a little inter-team interaction going on even real time during the seminar today. It's a real pleasure to have Steve speak to us. He has been working on some very exciting things that we're going to hear about. To give you a little bit of background on Steve, he is a professor of oceanography at the University of Rhode Island, has a bachelor's in geology from Stanford and master's and PhD in geological and geophysical sciences from Princeton University. He has been working for some time on studying life in extreme environments, particularly life in subseafloor sediments and biological controls on the chemical evolution of the ocean and atmosphere. And he has recently made a field expedition to the South Pacific Gyre, which we're going to be hearing about today, in a talk entitled Microbial Activities in Deep Subseafloor Sediments. And Steve, I'll turn it right over to you.
Thank you very much, Carl, for the warm introduction, and thank you for inviting me to have this opportunity to speak with all of our friends at NAI again. Carl has already read my title to you, so I won't repeat that. The people I've listed as co-authors, some of you will know as members of our team. These are team members whose work is directly related to what I'm going to present today. So this is a subset of the membership of our NAI alumni team. The two images I've put up there are photographs of the two ships that we've used for most of our expeditions. The lower photograph is a night photograph of the Gyres Resolution, the international drill ship taken in San Diego Harbor. And the upper photograph is the Roger Revelle, which is part of the university shared fleet of the United States. There's ships in many different oceanographic institutions. There's one at the University of Rhode Island. There are a couple at Scripps, plus some other special things at Scripps, and there are others scattered around the country. This particular one is a Scripps ship, and we used it because we were working in the Pacific. I'm going to start by drawing your attention to the blueness of the water there. I hope that you all have rooms dark enough to see this. This is a photograph of the surface ocean in the South Pacific Gyre. It is the clearest water in the world, the clearest natural water in the world. The deep chlorophyll maximum there is a little over 160 meters beneath the sea surface. The water is a beautiful periwinkle blue that actually edges toward violet. That's how clear the water is. You're not going to see that color of water anywhere else in the world. The topics I'm going to discuss today are listed against the backdrop of that water. I'm going to discuss life in subseafloor sediments, starting with a focus on biomass and moving on to activities and sort of categories of activities that are present, then activity per cell, a little bit about community activity, then I'll focus on some examples of effects on the surface world, which some of you can think of as potential biosignatures on a planetary scale. And then I will close with a little bit of speculation about potential food sources for subseafloor life. This first image here, if you go down right there, you see a glowing green dot. That's a microbe from a couple hundred meters beneath the Arctic seafloor. This particular sample was taken and photographed by David Smith, who was a member of our team. The reason that dot is glowing green is that it is stained with cybergreen, which preferentially binds to DNA and then causes fluorescence. So that is essentially a microorganism. These little glowing yellow-orange dots are from another cruise of ours in this case 320 meters beneath the seafloor in the equatorial Pacific. And this is a figure from Axel Schippers, who's one of our close European collaborators. These are stained with a different stain that binds to RNA. And this particular stain is specific to bacterial RNA. So what we're seeing here is that what would qualify as a glowing green dot is also a glowing yellow dot, because there's enough RNA in it to flash, which implies that this organism was recently active, because RNA has a very short shelf life. That's going to have some bearing on what we see next. The standard estimate of life's subsurface biomass, if you look up here, you'll see that the standard estimate by Whitman and colleagues, PNAS 1998, infers that 98% of life in the oceans lies beneath the seafloor in the sediments. You contrast that with the continents here, where we see about 20% is inferred to be beneath the surface of the continent. And then the global estimate essentially infers that a little over a third of life on Earth lies beneath the seafloor. Or I mean, beneath the surface of the world. Most of that is beneath the seafloor. So the standard estimate is essentially that a third of life on Earth lies beneath the seafloor. That estimate is based on the data from this paper, which is a paper by John Parks and his collaborators in 1994, where they presented cell counts from 5 ocean drilling program sites. And noticed that they, to a first approximation, all had roughly similar numbers of cells at every depth. So there's basically 2 assumptions being made then in Whitman's estimate. The first assumption is that the body counts correspond to living biomass counts. In other words, that all of the bodies counted are alive. And the second assumption is that those 5 sites are characteristic of the world as a whole, the ocean as a whole. We saw some clues in 2002 that they probably weren't characteristic of the ocean as a whole. That standard estimate, this is Parks et al. 2000 line, but it's not radically different than their 1994 line. The cell counts from our crews at these sites in 2002, you see that as we move offshore into the equatorial Pacific, which is still a nutrient-rich upwelling zone of the ocean, as we move offshore, the counts drop off. So the average counts seem to be characteristic of margin environments, like we see here, but perhaps not characteristic of the seafloor as a whole. So let's move a little bit further into this. This is data from Axel Schippers' paper that I showed you the figure from a short while ago. What you see here, basically, these are acridine orange direct counts, like cyber green. Well, acridine orange is actually a more complex stain. It binds to both DNA and RNA. But to a first approximation, since DNA is typically present long after RNA, we can treat this as a measure of the cells that contain DNA. And then this card fish stain here, this is the one that binds to RNA alone. So what you see is that roughly 10% of the cells on average that are counted with the DNA stain have enough RNA to flash with the RNA stain, which suggests that on average approximately 10% of the cells may be active and alive. Now the number that's alive may be even greater than this, but it sort of depends on where you draw the live-dead boundary. They don't have enough RNA for Axel and his collaborators to see, but it's conceivable that something could be alive, eking out an existence at some level that we can't identify. So the easiest way to interpret this is to say that conservatively, about 10% of the counted cells at those sites are alive, and perhaps more are alive. You can see this has big implications for the amount of biomass on Earth, because it can pull down by an order of magnitude, potentially, estimates of subseafloor biomass. Okay, now we're going to go to the South Pacific Gyre. I just got back from there in early February. So you're seeing shipboard data. These are counts by a former postdoc of ours, now a faculty member at the University of Potsdam, Jens Kallmeier. This is the ODP database that defined the line we saw before. This is a log-log scale, incidentally. All of these plots are log-log scales. And these black symbols are the data from our sites in this red box. So this site right here comes closest to the ODP data. That's located here on the northern edge of the Antarctic convergence zone, which is a major upwelling zone. So there's more organic matter raining down there than anywhere else here. Now, where I told you is the clearest water in the world is right here at Site 7. This base map is a site of sea surface chlorophyll. So all of these green areas are upwelling areas, and as you move to darker and darker colors, we're moving to lower and lower chlorophyll concentrations. The concentrations actually decrease here. This is essentially grams per square meter of sea surface. And our range here is from 10 to 0.01. So they're ranging over 4 orders of magnitude. And what we're seeing here is at the 0.01 end. What you see up here at that little tiny red area is at the 10 end, and what you see in these green areas is the 1 end, or in the middle at 1. So what we're seeing is that everywhere in this red box falls over here, with 3 orders of magnitude fewer cells than the average for all the previously counted sites. So what's this telling us? This is telling us that the standard estimate relied on sites that are in fact not characteristic of the seafloor as a whole. And that for all practical purposes, anywhere in the turquoise through black zone here has 3 orders of magnitude fewer cells than Whitman assumed the seafloor to hold per cubic centimeter. So let's back up a little more. When you look at the ocean as a whole, you see that in fact most of the ocean is in that range of turquoise through dark blue. So effectively then, the vast majority of the seafloor has somewhere in the ballpark of 1,000 to 10,000 cells per cubic centimeter. I shouldn't say the seafloor actually has about 1 million cells per cubic centimeter as opposed to 1 billion. And then once you're down in the subsurface, you're looking at about 1,000 to 10,000 cells per cubic centimeter instead of the million or so that Whitman assumed. So setting aside the issue of whether the bodies are live or dead, what you see is that the size of the subsurface biomass on Earth is actually far lower than the standard estimate. But don't lose heart, because as we'll see later in this presentation, they still get to matter. So we're going to speak about rates, about kinds and rates of activities now very, very briefly. There's been a lot of work in the last few years looking at microbial processes in subseafloor sediments. So we now know that a lot of classic microbial reactions occur in the subseafloor, many of which were known to occur in shallow marine sediments in the past. Sulfate reduction, iron, manganese, nitrate, and oxygen reduction all occur in different subseafloor environments. Carbon oxidation occurs almost everywhere. We may see an exception in a little bit. And anywhere where carbon is being oxidized, there are also intermediate products of metabolic pathways produced and destroyed, including ammonia, acetate, lactate, formate, hydrogen, methane, and somewhat surprisingly, ethane and propane. So some of the biological products that are created in these environments are actually historically thought to be abiological, meaning the ethane and propane. I'm not going to get into evidence here for why we think they're biological, but I'd be happy to answer questions on it. So this is sort of a standard model for what happens in anoxic sediments, whether on land or in the sea. The idea is that you start with organic matter. It's fermented by fermentative bacteria. The products of fermentation are then used as a food source for various reductions, manganese, iron, and sulfur reduction. Those redox activities constitute respiration, and the products of those reactions are CO2, which is the oxidized product of what started out as food, and manganese, dissolved manganese, reduced iron, and hydrogen sulfide, which are the reduced products of what started out as oxidants. And the evidence that these processes occur in the seafloor takes the form initially at least, of profiles of dissolved chemical concentrations. So you see here, sulfate is at a maximum at the top of this chart and at the bottom of this chart. The top is the seafloor. The bottom is the sediment-basalt interface, where the sediment lies on the oceanic crust below. And sulfate is coming into the sediment from the crust below and from the ocean above in dissolved form. In both cases, seawater is the ultimate source because seawater flows through the basalt. And then the concentrations decrease as you move into the sediment package, meaning that sulfate is being reduced. So the concentration is declining. In contrast, a standard product of respiration, what we call dissolved inorganic carbon, or total CO2, which is simply the sum of CO2, carbonate, and bicarbonate, shows the opposite pattern. It's at its lowest value at the top and bottom, where it's leaking out, and its highest concentration in the middle, where there is net production, just as there's net loss of the sulfate there. So that provides us with direct evidence that these processes are occurring. We can actually use those concentration profiles to calculate net rates of activity, where we use essentially the chemical's diffusion coefficient, the concentration gradient, depth, a measure of porosity at sites that sediment very rapidly, sedimentation rate. And we use something called formation factor, which is a proxy for tortuosity. Tortuosity is just the deviation from straightness, if you will, of the path that the chemical must follow as it moves through the sediment.
No, it's
don't call him.
So we've got somebody else live. So formation factor is actually a measure of...
It's a problem they're having.
I think Indiana or someone is alive. I'm not sure who.
I think it was Ames.
On the Ames, okay.
Yeah.
All right. So formation factor is a measure of the electrical path through the sediment following the seawater, and the chemicals follow the same path. So I'm not going to go over this in detail. I'm just going to wave a sort of a little red wand, if you will. But all of these products of respiration peak in the middle of the column, these anoxic sediments. So we have methane, ammonia, DIC, all peaking in the middle of the column. And then reactants, like sulfate, all decline or even disappear going into the column. And that's telling us multiple processes are occurring deep in the sediment everywhere. So if we take the results of those flux calculations, we can calculate how much electron acceptance is done by these different processes in different regions. So I'm just going to highlight the bold-faced things here. In the marginal sites, like along the Peru margin, where the cell counts resemble the traditional ODP counts, the dominant process is sulfate reduction. Then as we move into the progressively lower activity regions of the ocean, move out into the equatorial Pacific, sulfate reduction still dominates at the most active sites. But then metal reduction becomes increasingly important. So at the lowest activity site we visited in 2002, the dominant process, the dominant electron accepting process, is manganese reduction. Now if we go to the sites we visited in December and January of this winter, we see that the sediments in fact aren't anoxic. They're unlike any sediments people have ever cored deeply into and looked for life. What we see there, this is our little site at the Antarctic convergence down here. There oxygen disappears in the first meter of the sediment column. So that's a typical sediment in the sense of what people expect. But all of the other sites, the sites that were in that red box where the cell counts are so low, are over here. So there's 150 micromolar or more oxygen throughout the sediment column. That's following along this arrow here. So that's essentially, again, in the region of turquoise, dark blue, and black chlorophyll content in the overlying ocean. And nitrate is always above or at deep water concentrations. It's a little bit of nitrate produced by oxidation of organic nitrogen in the shallow sediments, and that's what makes it a little high. And there's no nitrate reduction below. So what we're seeing here And sulfate doesn't change at all anywhere. In terms of carbon equivalents per year. It's in the range here of 10 to the minus 16 to 10 to the minus 18 moles per cell per year. In coastal sediments, you see numbers more in the ballpark of 10 to the minus 15. And in pure cultures, it's typically in the range of 10 to the minus 13 to 10 to the minus 11. So this is a sort of odd claim, because essentially we're claiming that subseafloor organisms, on average, are respiring at much, much lower rates than anyone's observed in the surface world. Now, you have a couple of possible ways of explaining this. One, the claim is true, that these things are respiring very, very slowly. Two, it's conceivable that we've missed some huge, huge source of respiration somehow, orders of magnitude. And three, it's possible that only a fraction of the cells counted are actually alive and respiring. So if we were to take, for example, the shipper's estimate that 10% of the cells are alive, but perhaps no more, then all of these could be brought up in order of magnitude, and they'd be 10 to the minus 17 to 10 to the minus 15. The low end, they'd start edging toward the shallow sediments. Okay. So what we decided to do was to see whether we were missing some obvious source of, or even inobvious source of respiration. And instead of assuming, as we did, that sulfate reduction was the dominant process, as we did in our 2002 paper, we said, let's look at the amount of DIC produced, the amount of CO2 produced by respiration of organic matter. So there are two things that we have to consider here. The first, we have to consider the flux of dissolved inorganic carbon out of the sediment. Second, we have to consider the amount of carbon that precipitated out as a carbonate mineral within the sediment. So that's, in a sense, a hidden term. We can calculate that second flux by looking at the calcium and magnesium fluxes into the sediment. So it's quantifiable. And when we do that, we can calculate carbon oxidation rates at different depths, take the cell counts, and calculate the carbon oxidation rate per cell. And you see that using this carbon-based approach, we're still getting average values for carbon oxidation of around 10 to the minus 17 moles per cell per year. So this essentially means that we aren't missing a very big source of respiration somewhere in the system, and that either most of the cells are not active or they really are breathing at phenomenally slow rates. But again, if 10% of the cells are active, as Shippers says, then they're still less active than anything known in the surface world. So just for comparison, here's the total flux assumed by looking at the oxidation, the reduced products, if you will, which is the way we've done these calculations in the past. And this is the flux from the CO2 mass balance, which is looking at the oxidized products. You see that the two numbers are nearly identical. You'll also see, well, let's go back there. Okay, I transferred this as a screen capture a few minutes ago and lost my fields. In here, this is the amount of energy that Price and Sowers infer is required just to keep the organisms ahead of racemization of their amino acids, with the isomers reverting over time. The traditional estimates of maintenance energy up around here. So coastal shallow sediments, organisms were already operating near the inferred maintenance energy necessary for life. And what we're seeing here is that these microbial communities are actually being maintained at far lower rates of respiration than theoretical estimates of maintenance energy have allowed in the past. So, if nothing else, the physiology of microbial maintenance isn't yet well understood. I'm going to focus very briefly here on something that one of our graduate students did part of her thesis work on. essentially there's thermodynamic cooperation going on in these systems. I'm going to breeze through this fairly quickly, but what you see here, this table essentially calculates how much carbon is oxidized by a given process, so sulfate reduction, iron reduction, manganese reduction, and how much carbonate is precipitated as a result of the changes in the alkalinity due to that process. And then we look at essentially not just then the mass balance of say, the reductions and the carbon oxidation, but also effectively the mass balance of the carbonate precipitation and the reductions. And when we do that, we find that if you were to take this DIC flux and this carbonate flux and sum them, it'd be nearly equal to the sulfate reduction rate. So those two numbers are nearly balanced. So from a DIC or from a carbon production perspective, given the uncertainties of our approach, you don't need to call on anything but sulfate reduction for this system. But it turns out when you reduce sulfate, you actually dissolve a little carbonate. You don't produce it. And here it's being produced. So what we're seeing is that the carbonate precipitation is too high to be explained by sulfate reduction. So the simplest way to get lots of carbonate precipitation is to add a little iron reduction along with the sulfate reduction. Because it turns out iron reduction, you end up precipitating a lot of carbonate. So what this is telling us, what this balance is telling us effectively, is that iron reduction and sulfate reduction are co-occurring in these sediments. This is a little unexpected for some people because usually in the standard depth. What we're seeing here is that throughout a sediment column that was deposited over about 15 million years, iron and sulfate reduction are occurring together. So the iron hasn't outcompeted sulfate. Instead, both of those two are co-occurring. Let's take a closer look at that. Essentially, what we believe is that in a sense, these communities constitute an underground economy where there's an invisible hand of thermodynamics keeping the system in play. If you look here at this figure, these are the in situ free energies of acetate oxidation using hematite reduction, methanogenesis, this is basically just acetate disproportionation, and sulfate reduction. And you see that all of these free energies are in the range of around minus 50 to minus 20 kilojoules per mole of acetate. The minimum free energy of this reaction is typically assumed to be somewhere in this region. So these are very low free energies, and you see they're also pretty constant throughout the sediment column. The ones where all of the products and reactants are dissolved, methanogenesis and sulfate reduction, they're nearly constant. Where we're inferring a mineral, it's more variable. This could mean that the free energy of that reaction truly varies from depth to depth, or it could mean that different iron-containing minerals are being used at different depths and the free energy is more nearly constant. The standard free energies of these reactions vary considerably. The standard free energy of the iron reduction is somewhere around minus 1400 kilojoules per mole of acetate. The standard free energy of the sulfur reduction is roughly an order of magnitude less than that, but still well off the charts here. So what we're seeing is that all of these processes are operating fairly close to a biological minimum, and they're relatively constant throughout the column. So that implies that there's some sort of thermodynamic homeostat, if you will, with the thermodynamics sustaining this process. I don't have a slide to explain this, so I'll just describe some examples of how this might work. As sulfate reduction proceeds, you're going to build up dissolved sulfide, and you're going to build up dissolved CO2. The products of thermodynamic reactions, as you probably all know, poison the reactions. So if you allow product concentrations to build up, the free energies of the reactions go down. So if you let your dissolved iron concentrations go up, the free energy of iron reduction goes down. If you let your dissolved sulfur concentrations go up, the free energy of sulfur reduction goes down. Very conveniently, however, if you have reduced iron and reduced sulfur produced in the same system, they precipitate out together as iron sulfide. So the coexistence of these reactions allows their products to scrub each other out. So effectively what we're suggesting is that, in a sense, these organisms are cleaning up the respiration products of each other and allowing the reactions to proceed. Methanogenesis, similarly, when methane is produced, that methane can then be used as a food source by the sulfate reducers and the iron reducers. So the implication is that on the time scales of millions of years over which these ecosystems are operating, these systems reach a more or less stable point where these processes that are traditionally believed to compete can in fact coexist because they're cooperating at some level. So I'm going to briefly speak then about some of these processes in terms of their planetary implications. I suspect that everyone here today recognizes that methane is a significant greenhouse gas. It has much lower concentrations than CO2, which we see in the news a lot, but its effect on global warming per unit of atmospheric constituent is much greater. So one consequence of this subsurface world is that the methane produced by these microbes can, and probably on long time scales, does significantly affect Earth's climate. For example, the really big global warming event of 55 million years ago is generally inferred to have been the result of subseafloor methane being disgorged. And that methane is principally created by methanogenic microorganisms beneath the seafloor. For those of you who are less climate focused and more resource focused, these subseafloor methane deposits are actually the largest untapped fossil fuel reservoir on Earth. So here's an example where these relatively slow activities by these subseafloor microorganisms have global implications. I'm going to point to another example that, to the best of my knowledge, is not publicly discussed except by me and hasn't actually been published. And that is that ultimately subseafloor sulfate reduction affects atmospheric CO2. Or to put it a little differently, the process of oxidation of sulfur, reduced sulfur, and reduction of oxidized sulfur affects atmospheric CO2. So if we increase, our current concentration of dissolved sulfate in the ocean is about 28 millimolar. If we increase the concentration by 1 millimolar, the effect on ocean alkalinity will drive up atmospheric CO2 by a factor of 3. So if we decrease the concentration by 1 millimolar, it'll pull atmospheric CO2 down to nearly 0. This calculation is effectively, in terms of subseafloor, in terms of sulfate reduction, this calculation is relative to, say, carbon oxidation by free oxygen instead of by sulfate. Effectively, oxygen has no effect on reduction, and this would be relative to that. You get a cleaner relationship if you were just to look at pyrite oxidation. If you oxidize more pyrite, you will drive the absolute concentration up by this without having to worry about any respiration effect of the CO2. So the basic point is that this is a very, very strong lever for changing the composition of the atmosphere and ocean. To put it in sort of a text form, the removal of sulfate from the ocean by precipitation of iron sulfide is equivalent to removal of a strong acid. Similarly, the addition of sulfate to the ocean by weathering of iron sulfide is equivalent to creation and addition of a strong acid. And this may actually serve to modulate Earth's climate on long time scales. For example, when we have warm climates, we have low dissolved oxygen in the ocean because warm water holds less gas than cold water. Consequently, less organic carbon goes to oxygen reduction, more goes to sulfate reduction. and you can increase the iron sulfide burial in the deep sea, which in turn can pull down atmospheric CO2 and cool the atmosphere and ocean. Now this is a process that's not going to save us on the timescale of fossil fuel burning because the rates are relatively slow. This is a sort of process that plays out on timescales of hundreds of thousands to millions of years. Now I'm going to briefly touch on something that's of particular interest to some in the NAI community, particularly people at the Indiana team who've done a very nice job working with deep gold mines. We're going to look at something that does not appear to be analogous, but in fact may be in some ways analogous to what they're looking at there. The principal sources of food, or electron donors, for subseafloor life are the burial of photosynthesized organic matter, the radioactive splitting of water, potentially alteration, that would be oxidation or reduction of minerals, and the thermal creation of organic matter or other reduced compounds. So the sorts of sediments that my team principally focuses on, we can ignore both mineral alteration and thermogenesis. These are very cold sediments, so thermogenesis is not an issue. And in the lowest activity sediments that we look at, everything is oxidized, so there aren't any real minerals around to be oxidized, because they've all been oxidized in advance. So instead I'm going to focus on burial of photosynthesized organic matter. Here's a pretty picture from Steve Pesch and colleagues showing microbes growing on a carboniferous black shale that he's exposed in a thin section, just showing that if you can get organic matter to them, they'll use it, even if it's hundreds of millions of years old. And this is a little schematic that just shows the process of radiolysis of water, where you have decay of uranium, thorium, potassium, creating alpha, beta, gamma radiation. And that radiation, some subset of it strikes mineral grains and essentially just goes into leaving fission tracks, if you will. But any of it that gets out of the mineral, out of the rock, and into the surrounding water will split the water. There's about 30 of these radiolytic reactions, but the stable products of them are water, again, hydrogen, form of dihydrogen, and the oxidants O2 and H2O2. When we do mass balance calculations based on burial rates of organic matter and the reduction rates I showed you earlier, the relatively high activity sites that we went to in 2002, these Peru margin sites and these open Pacific sites, essentially burial of organic matter is providing enough food to sustain the net rates of sulfate, metal, and nitrate reduction that we see at those sites. You see that these numbers are generally in the same order of magnitude, or the second number is higher than the first. So that's telling us there's enough organic matter around to sustain those processes. So at those traditional anoxic sediments, the principal source of food is basically buried organic matter. So let's take a little closer look at how much energy might come from the alternative process, the radiolysis. So these are the same sites ordered from high to low energy. What we've done here is we've calculated how much radioactive radiolytic hydrogen would be created given the uranium, thorium, and potassium content of the sediment and its porosity. We compare that to the carbon-fueled respiration that you saw on the previous slide and get a ratio of the two. And what you find is that at the ocean margin sites, like 1230, less than 1% of the energy or the food that fuels that system comes from radioactive splitting of water. But as we move offshore into lower and lower activity sediments, till we get to the Peru Basin where manganese reduction is the dominant process, radioactive splitting of water, at least based on our calculations, appears to provide a little over 10% of the amount of energy that the buried organic matter is providing to the system. So what is another way of looking at this? That's a site where we had around 100,000 cells per cubic centimeter of sediment. So if we were to go to a site where we simply had radioactive splitting of water as our only source of energy and we had the same amount of radiolysis, we'd be able to support around 10,000 cells per cubic centimeter. And when we go to the gyre, this is sort of right here. You see that oxygen didn't disappear. Our estimates of of net activity are 3 orders of magnitude less than in any sites where people have looked before. So it looks like this is a region where if the sediments are as radioactive as those we've seen at the other sites, radiolysis may in fact be the principal source of food for the subsurface ecosystem. Now it's important to bear in mind that radioactive splitting of water does not add to the net processes that we see in these profiles, because oxygen is created at the same time as hydrogen. So you're creating oxidant at the same time you're creating reductant when you have radiolysis of water. So in a sense, we can use these profiles simply as a measure of the organic fueled respiration. What you're seeing is that organic fueled respiration nearly stops within the first meter of the sediment and then goes to essentially zero, or very close to zero, in the heart of the gyre. And if you think back to what I showed you before, you'll remember that concentrations, sorry, I'm jumping ahead to see if I had another slide. You'll remember that concentrations of cells in this region were 3 orders of magnitude less than the average. So we're seeing about 1,000 cells per cubic centimeter in most of these sediments, the range of 10,000 to 1,000. And so one of the things we'll be pursuing over the next year is testing whether or not those 1,000 to 10,000 cells per cubic centimeter are likely to be supported principally by the radioactive splitting of the pore water here, instead of by the buried organic matter. And with that, I'll bring this to a close and answer questions.
Steve, thank you very much. That was quite fascinating. I think I'll ask the first question, and if others would raise their hands in the meanwhile on the chat side of WebEx, we'll then go around the loop. This whole idea of thermodynamic cooperation between different groups of organisms so that you have metabolisms coexisting that you wouldn't have normally thought would coexist is sort of fascinating. It reminds me a bit of the anaerobic oxidation of methane, where perhaps that's also going on as well. So given that you have another very prominent example that may actually apply to a very large segment of the seafloor surface, I'm wondering how much do you think, might you speculate, remains to be discovered? Are we going to discover a lot more of these thermodynamic cooperations that yield metabolisms or coexistence of metabolisms that we wouldn't otherwise have expected? And should this kind of thermodynamic cooperation open up our ideas about what might be possible on another planet, let's say Mars?
That's a good series of questions, and I'm going to start by saying you're right. This in many respects resembles the situation with anaerobic oxidation of methane. In fact, work that Torrey Holder did, particularly in his thesis, provides a sort of guide, if you will, to this sort of behavior, where he first estimated free energy yields in relatively shallow sediments for that process. So you can think of this in a way as an extension of that idea into other processes, into deeper sediments in much longer periods of time. In a way, the AOM example and the example I gave are both extensions of an older recognition, which is that organisms feed each other. You know, for example, it's well known that the sorts of organisms that undertake redox activities like sulfate reduction and iron reduction in anoxic sediments rely ultimately on fermentation of organic matter for their food in sort of typical swamp sediments. So you can think of this as a way of formalizing that older microbiological recognition and putting it in sort of a thermodynamic basis. And I think we will see a lot more of it as people begin to think in that more explicitly thermodynamic way. One of the things that, I'm not going to get into too much detail here, but what we've seen in our more recent calculations are that processes that don't even occur in the surface world, because the free energies don't allow it, actually occur in the subsurface world, because in situ free energies are very different in the subsurface than they are in the surface. And so there are interactions in the nitrogen cycle, for example, that you won't see in the surface world that appear to occur in the subsurface. And I suspect that if people were to move back out of the subsurface into the water column, particularly in anoxic sediments, we would see these rules apply there as well. And by extension, if we were to search for these processes on other planets, I suspect, or other bodies, if there's life there, we would find that they would follow similar rules if the environments are stable enough. For example, if there is an anoxic community beneath the ice of Europa or beneath the surface of Mars, one would expect that over relatively geologically short periods of time, you'd reach these sorts of community equilibria, if you will. And one of the things that there are complications in this that potentially make it of even deeper interest. For example, you can imagine a system where iron reducers oxidize sulfur, and then sulfur reducers oxidize an organic matter, and the iron reducers don't do the organic matter directly. Well, in that case, you create an electron shuttle, if you will, where you can actually sustain more organisms than with the same amount of energy, but the iron reducers and the sulfur reducers both going for the organic matter. We've not observed that in any of these systems. We haven't looked for it either. So it's possible to imagine ways of not only sustaining a richer community metabolically, but sustaining a community that uses its resources much more efficiently and sustains a larger biomass. And I'll stop there.
Okay, we have a question from Penn State.
Hi, Steve. Jim Casting. I thought the most provocative thing you said was that oceanic sulfate controlled atmospheric CO2 and climate. And when I give my talk on climate, I usually emphasize that CO2 is more controlled by the inorganic carbon cycle, the carbonate silicate cycle, and that the organic carbon cycle can't really do it because it buffers oxygen. What are your thoughts on that?
And that's the usual perspective, and it's a very good one. The easiest way to put the control here is that to step aside from the one problem with sulfate reduction as a control is you're creating DIC at the same time that you're removing sulfate. So in a sense, you're replacing an anion with another anion, and so the effect on it's more nearly a zero-sum game there than I presented it at colloquially. But it's a more pure case where you look at, say, pyrite oxidation, where you start with iron sulfide. If you oxidize that, and you get an oxidized iron that doesn't go anywhere, and you get an oxidized sulfur that goes into the aqueous system, and that's a strong acid, and it's going to drive off CO2. So the cleanest way of imagining this operating would be to essentially just change your rates of precipitation and oxidation of metal sulfides. And through the alkalinity system, it'll have an effect on atmospheric CO2.
Lee Kump is here. Lee, do you want to add anything?
Hey, how you doing? Well, I mean, I guess part of on the weathering side, it depends what happens to the protons, right? And so there's a lot of interest I've been seeing these days in looking at pyrite-induced weathering of silicates, for example. And so if those protons are taken up during silicate weathering before they ever make it to the ocean, then you get sulfate going in for sure. But the protons are actually being neutralized on land, and so then it goes back into Jim's camp. So I think part of it on the pyrite oxidation side depends on the fate of those protons. Now, on the flip side, the net sulfate reduction and barrel of pyrite, there, that's sort of like a proxy for organic carbon barrels. So there, I think you have a CO2 driver associated with that barrel of pyrite. But it does have implications for oxygen as well, and so you need to think about that. Just like you do for any CO2-driven thing that is based on organic carbon barrel, you have to worry about what's the fate of that oxygen. Did you drive up an O2 pulse with the CO2 drawdown.
Yeah, as you're well aware, as both of you are well aware, people have often looked at sulfur in terms of oxygen sinks. It's sort of an extension of the classic burner type approach. I find it sort of interesting that people rarely look at the alkalinity side of it. But.
Okay, we have a question from Carnegie. Carnegie, are you there?
Yeah, hi. Fustukos from Carnegie. I have a question regarding DOC. Have you measured DOC in these sediments?
We do measure DOC. I can't right offhand remember any numbers for you. We measure DOC, and sometimes we also actually measure classes of DOC. For example, carbohydrates, amino acids, etc., in dissolved form. The DOC, of course, is always dissolved, but I'm trying to clarify the carbohydrates and amino acids. We look at those in dissolved form also. Do you have a question specifically about the DOC?
Sorry, I'm having issues with my sound, so it's okay. It's okay. I kind of don't know what we're talking about.
Okay, we have a question from Indiana.
Schmillmann, Indiana University. If you want to find out what is the contribution of radiolytically provided energy in contrast to organic matter in deep sea sediments, have you looked at horizons where you have a dilution of sediment by tephra? In those intervals, you might expect less organic material, but more radiolytic energy. And do you have maybe a blip in bacterial abundances at the margins of these tephra layers or in this association?
We haven't specifically looked at this question of tephra versus normal sediments, and the main reason for that is that the concentration of organic matter as you go from the continental margins to the middle of the gyre varies by an extraordinary amount. You know, we're talking orders of magnitude, and so essentially you can take a grab sample of sediment from the middle of the gyre and have an essentially organic free sample for comparison. And this allows us to use thousands of kilometers distance for controlling our system instead of worrying about centimeters. I don't know if that answers you. I'm answering your question by saying no, we haven't looked at the tephra case, but that's because we have a very big geographic case that we're looking at.
Thanks a lot.
Yeah.
Okay, we have a question from Goddard.
Hi, Steve. It's Mike Muma here.
Hi, Mike.
It was really a nice talk. I enjoyed that. I wanted to ask you a quick question about the balance of reductants to oxidants in the radiolysis-dominated regime.
Yeah.
Wouldn't that depend strongly on the tortuosity of the medium, since molecular hydrogen is much more mobile than molecular oxygen and can squeeze through tighter spaces, if you like? And I would think that it would escape more readily and leaving the medium relatively rich in oxidants as opposed to...
Yeah, it depends on... you're absolutely right. The hydrogen should escape more easily. In the circumstances we're looking at here, we're essentially looking at sediment thicknesses. On this recent cruise, the deepest sediment we cored was about 8 meters, and the deepest sediment piles that we cored into are around 130 meters. Okay, so we're essentially looking for diffusive distances that are in the shortest distance, going say halfway through the sediment column to the surface or to the basement below, could be as little as 2.5 meters, where the sediment's really thin. Or in another system, it could be as much as 70 meters. So if you've got a dead system where the hydrogen or the oxygen aren't being utilized, you would expect your steady state concentrations of those two products to be different because the hydrogen's escaping more readily. You'd have an excess of oxygen, if you will. But in a system where you've got organisms cropping it, typically they crop the hydrogen, in timescales of minutes or less.
Wow.
And the distances over which the hydrogen must diffuse is, again, meters to tens of meters. So that will take many years to thousands of years.
Has anyone actually done any kind of measurements of net gas escape from these media to identify, use that as a marker for the kinds of biology that's going on, whether it's driven radiolytically as opposed to by other mechanisms?
Well, we took samples on our recent cruise. We measure hydrogen routinely. Yeah. And what we found was that hydrogen is always below our detection limit. And our detection limit is typically 2 or 3 orders of magnitude lower than the concentration you'd expect if the sediment is as radioactive as the sediment we've looked at in the past and the system is dead. Okay. We also took samples for measuring helium concentrations to get a measure of the alpha flux out of the sediment, if you will.
Right. Right.
And at steady state, those helium concentrations, the helium flux should be equal to the helium bombardment of the water, because obviously any alpha radiation that didn't get out of the mineral grains won't be in the water to diffuse out. So we're hoping to use that as a check, if you will. Both of those are variations on things that the Indiana-Princeton team has done in their deep mines. One difference being that effectively they're measuring concentrations there, and they don't have the advantage of depth that we have, where we can actually calculate fluxes directly using transport models. But we're measuring the same products that they measure, essentially.
Thank you very much.
Okay. Well,
Okay.
thank you, Mike.
It looks like
Go
that's
ahead.
all. That's all the questions. I just wanted to make one other comment, and if anybody has a last-minute question, please raise your hand while I'm doing so. Otherwise, we'll sign off. And that is that one of the things that we have been doing is working to improve the quality of our archives of these seminars and others. And so I just want to remind you that all the director seminars are archived. The archives are available on our website. One of the things we have not had archived is the video feed. What we've had is the audio feed and the WebEx, or at least the slides, I should say. And we now have an archive that includes the video feed, so you can see Steve's handsome face, hopefully, when we archive this. We will maintain both options, however, because the archive version that has the video feed is a much larger file size, and it's going to be inconvenient for anybody who doesn't have ready access to a fairly broadband link. So we will have both options available, but I just wanted to let you all know that. And let me just ask if there are any last questions from anyone around the net.
I think Arizona has a question.
Arizona, go ahead, please.
Hi, Dr. Daunt. It's Joe Russell.
Hi, Joe. How are you?
Good. I was just wondering, it's sort of a method question, really, is how do you, since you're taking these core samples from the ocean, how do you make sure that it's not contaminated by ocean water? And obviously there's a lot of microorganisms in ocean water. So exactly how do you, do you account for that? Do you have to account for that at all?
Yeah, that's a very good question. When we're drilling, it's easier to do than when we're simply piston coring, as we were on the most recent cruise. What we do when we're drilling is we do two things. One, we put a little bag, a Whirlpack bag, with fluorescent spherules into the cutting shoe. So when the piston drives the core forward, it ruptures the bag, and those micro spherules all just stream up the sides and into the core. I'm not real fond of that technique because it tends to disturb the core. But microbiologists like it because they can see little glowing beads in their microscope slides. The technique that we use that's quantifiable and more precise and more accurate, in a sense, except as a true-false indicator, is that we put a very easily measured chemical into the drilling fluid and circulate it through the system. It's a perfluorocarbon tracer that we can measure at the PPT level. And because we know the rate at which we're metering it into the drilling fluid, when we find it in the core, we know exactly how much contamination has occurred. And incidentally, the drilling fluid is surface seawater. So that's how we do it when we're drilling. Yeah, there are much less subtle approaches that people have used. For example, you can use the difference in chemistry between the surface water and the pore water as a proxy for contamination. But that's usually only picking out really gross contamination. So for example, you could use the occurrence of oxygen in anoxic sediments as a measure, or the occurrence of sulfate in sulfate-free sediment as a measure. But the perfluorocarbon tracer is much, much more sensitive. So essentially our detection limit for the PFT is equal to somewhere between 1 and 10 cells of contaminating microbe, which is about as low as anyone's gotten it, although there are hypothetically ways of getting it lower. Okay.
Thank you.
Thank you. I don't know if you can hear me.
You're welcome. Thanks for your question.
Okay, Steve, I think that's the end of the questions, so let us all thank you again for a really fascinating and stimulating and provocative seminar, and we look forward to hearing more.
Thank you very much, Carl. Thanks for